Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the animal science, that is the zoology part. My dear students, in the previous lecture, we have gone through amino acids and their different types, okay, like acidic, basic, neutral, polar, non-polar, different types of amino acids being discussed in the previous lecture. Today we are going to start about the polymers of amino acids known as proteins. So proteins, they are basically variously folded linear heteropolymers of amino acids. Okay, so proteins, they are heteropolymers. Okay, heteropolymers of amino acids. Okay, linear. The linear polymers of amino acids are called polypeptides. Linear polymers of amino acids is known as polypeptides. A protein may constitute of one polypeptide or many polypeptides, like one polypeptide, then it's monomeric, two polypeptides, then oligomeric, and then more than four polypeptides, eight polypeptides, and so on. Okay. So a protein have two or more polypeptide is called multimeric. It's called multimeric. Two polypeptides, oligomeric. One polypeptide, monomeric. So configuration of protein molecule, that is the primary configuration of primary structure we are going to discuss about. Okay. So according to this primary structure, this is the basic structure of protein. Amino acids are linked by the peptide bond and are arranged in a straight chain, form the primary structure of proteins. The protein structure is newly produced on the ribosomes, are primary structure and are highly unstable. The protein structure which are newly produced on ribosomes during translation, they are primary structure and they are highly unstable. Okay. So you can see the amino acid residue like serine, cysteine, tyrosine, glutamic acid, they make the protein. So this primary structure is known as what? This structure of the protein is known as what? The primary structure, and it makes the basic structure of the protein molecule, and it is highly unstable. It is further converted into what? The different other like secondary, tertiary, and other structures. Okay. So in this diagram, the primary structure of protein, especially the nitrogen and carbon, represents the first and last terminals. This is the last terminal, which is the amino or N terminus, and this is the carboxy terminus, carboxy or C terminal. Okay, understood? Now, secondary structure. So before moving to the secondary structure, we will discuss some more points for primary structure as well. Okay. The primary structure, it is the most basic structure, as I have already told you. It includes number of polypeptides, okay, which are sequentially arranged. In case of genetic, when we talk about the genetics or the DNA, So through translation or transcription, whatever the amino acid is formed, that is the primary structure. So please remember that the first amino sequence which is prepared by translation process is having the primary structure. Please remember it is having a primary structure which further goes, folding and multiple folding to form different other forms, a secondary or tertiary structure. The distance between two adjacent peptide bonds is about 0.35 nanometers. The distance between two adjacent peptide bonds. Distance between peptide bonds. Nano. Okay. Secondary structure. Secondary configuration. The protein molecule are spirally coiled in the secondary structure. They are spirally coiled in the secondary structure. Now the amino acids are also linked by hydrogen bonds. Okay. So along with the peptide bond, one more bond is present now. That is the hydrogen bond, which are formed between the oxygen of one amide group and the hydrogen of the another amide group. So please remember, in the primary structure only the peptide bond is there, only the peptide linkage is there. In the secondary structure, peptide bond plus hydrogen bond is there. Okay. So the hydrogen bond is formed between the oxygen of the one amide group and the carbon of the hydrogen of the another amide group. Proteins are insoluble in water and have fibrous appearance. This structure is of two types. Proteins, they are insoluble in water and they have fibrous appearance. This structure is of two types. Okay. The alpha helix. Alpha helix is a right-handed rotation of the spirally coiled chain with approximately 3.1 or 3.1 by 2 amino acids present in each turn. These are intramolecular hydrogen bonded. They are intramolecular hydrogen bonded between two amino acids of same chain present in the structure. Same chain present in the structure, like keratin, myosin, and tropomyosin. Okay. So proteins, they are insoluble in water and they have fibrous appearance, and this is having two types of structure. One is the alpha helix and the second is beta pleated sheet. So the alpha helix is a right-handed rotation of spirally coiled chain having 1.2 or 3 complete and 1 incomplete or partially complete amino acid present in each turn. So around 3.5 amino acids are present in each turn, because half of the amino acid it goes into the next turn. They are having intramolecular hydrogen bonding, means between the amino acids of same chain they are having hydrogen bonding. Hydrogen bonding, basically it exists like this. Like this is the amino acid RC NH2 C double bond O N H C OH RH. You can see this is the peptide bond, this one is the peptide bond, and the oxygen hydrogen bond. This is the hydrogen. Okay, understood? Yeah. So the alpha helix is having intramolecular hydrogen bonding between two amino acids of same chain. Examples are keratin, myosin, and tropomyosin. Keratin is a sclera protein which is fibrous, tough, and resistant in terms of digestion. Okay. So keratin is a type of sclera or scleroprotein which is present in spatially covering the body of arthropods and all. So it is resistant towards the digestion of acids and all. So there is abundance of cysteine or sulfur containing amino acid. Sulfur containing amino acid in the structure which gives the hardness to keratin. So keratin is a scleroprotein which is fibrous, tough, and resistant in terms of digestion. there is abundance of cysteine, that is sulfur containing amino acid in the structure, which gives the hardness to keratin. Beta helix or beta pleated sheet, the protein structure has a zigzag arrangement. It is having a zigzag arrangement. The protein molecules, they are held together by intermolecular hydrogen bonding. They are held together by intermolecular hydrogen bonding. Examples are fibrinoin, which is present in silk. So the alpha helix is present in tropomyosin, myosin, and keratin. Okay, keratin is a scleroprotein which is fibrous, tough, and resistant in terms of digestion. There is abundance of cysteine or sulfur-containing amino acid in the structure, which gives hardness to keratin. Beta helix or beta pleated sheet, it is having a protein structure which is having a zigzag arrangement. The protein molecules are held together by the intermolecular hydrogen bonding in case of beta helix or beta pleated sheet. Understood? Example, fibrinoin or silk. The alpha helix and beta pleated sheet. This is the example of alpha helix and beta pleated sheet. The hydrogen bonds between amino acids at different locations in polypeptide chain. You can see the hydrogen bonds between amino acids at different locations in polypeptide chain. This is alpha helix. It shows intramolecular hydrogen bonding, H bonding. But the hydrogen bonds between amino acids at different locations in the same chain. In the same chain. Okay, you can see different locations, okay, in the same chain. The beta pleated sheet, it is having intermolecular, I mean hydrogen bonding between two different zigzag chains. One zigzag chain is this, and the other zigzag chain is this. So they have hydrogen bonding in between, okay. Intermolecular hydrogen bonding. So intermolecular hydrogen bonding is present in beta pleated sheet, which is a type of secondary structure. The secondary structure is having the alpha helix and beta pleated sheet, right? There is another term which is known as the collagen helix, okay? So basically the secondary structure, it is of three types: alpha helix, beta pleated sheet, and collagen helix. Alpha and beta, they are the most simple designation of the secondary type of structures. Like the first and second type of secondary structures are also known as alpha helix. So alpha helix is the first type of secondary structure, and beta pleated sheet is the second type of secondary structure. Okay. Alpha helix, the polypeptide chain is called spirally, already explained. Okay, your right-handed helix is there, right-handed manner. At present, the helix is less regular, forming random coils. The helix is stabilized by hydrogen bonds between oxygen and carboxylic group of one amino acid residue and NH group of next fourth amino acid residue. Please remember, the hydrogen bonding takes place between the hydrogen bonding takes place between the amino acid residue and the NH group of next fourth amino acid residue, mean between the C terminus or carboxy terminus, CO group of first amino acid residue with the amino group of fourth amino acid residue. Okay. Actually, all the main chains CO and NH groups are hydrogen bonded. Alpha helical coiled secondary structure is found in several proteins like keratin, myosin, tropomyosin. Okay. Fibrinoin. Fibrinoin is having beta pleated sheet. Fibrin, blood clot, is having alpha structure. So fibrin is having alpha structure, and fibrinoin, which is silk protein, is having secondary structure. Okay. Then alpha helix and beta pleated sheet. These are the differences. Right-handed coils rod-like structure. In alpha helix, it is sheet-like structure. The hydrogen bonds formed within the polypeptide chain means intra, and this is inter. Hydrogen bonds formed between NH group of one amino acid with CO group of another amino acid. Okay, 1 to 4th linkage. Hydrogen bond between adjacent 1 to linkage. Okay. In between the neighboring NH and CO group of adjacent peptide chains, R groups of amino acids are oriented outside of the helix. Okay. R groups are directed to both inside and outside of the sheet. Alpha helix can be single chain. Beta helix cannot exist as a single chain, single beta strand. Alpha helix has only one type. Beta sheet can be parallel, anti-parallel, or mixed. Okay. Alpha helix prefers alanine, leucine, methionine, okay, and many more. Beta sheets prefers tyrosine, tryptophan, okay, isoleucine, valine, many more. So these amino acids, they prefer to be in alpha helix, like alanine, leucine, methionine, then phenylalanine, glutamic acid, histidine, lysine, arginine, and beta sheet prefers all these amino acids. Okay. Tertiary structure. Proteins in the tertiary structure are highly folded and form a globular appearance. So proteins in the tertiary structure, they are highly folded and they form a globular appearance. Okay. They are water soluble, form colloidal solution. So they are water soluble and they form what colloidal solution. They don't make true solution. This structure has following bonds. Peptide bonds are strongest bond present in the proteins. Okay. Mean if we conclude that basically the covalent bonding is the strongest in proteins, and covalent bonding is of two types. One is the peptide bond and the second is disulfide linkage. This is disulfide linkage. Okay. One is the peptide bond and the another one is disulfide linkage. So peptide bond and disulfide linkage, they are the two types of covalent bond which are the strongest in protein structure. Okay. Hydrogen bonds between hydrogen and oxygen of amino acid, they are weak in comparison to the covalent bonds. Okay. Disulfide bonds, the bond between sulfur and hydrogen group of amino acid, especially cysteine, which are the second strongest bond in the protein and stabilize the tertiary structure. So the first strongest bond is peptide bond, then the disulfide bond, then the ionic bonds, then the ionic bonds. Okay. Hydrophobic bonds, the bond between the amino acids that contain the hydrophobic side chains like aromatic amino acid. Okay. So aromatic amino acid, they have hydrophobic side chains and they make hydrophobic bonds. It increases the efficiency of packing. It increases the efficiency of packaging or compaction, consolidation. Ionic bonds. The formation of ionic bond between the two opposite ends of protein molecule is due to electrostatic force of attraction between them. So because of the distribution of charges, because of the dispersal of charges, these charges attract the two chains or the groups present in two same or different chains, and this is known as what? Ionic bond. So the strongest bond is peptide, second strongest is disulfide, then ionic bonds, then come the van der Waals forces of interactions. Okay, van der Waals forces of interactions, and then the hydrophobic bonds. Okay, understood? The first is covalent bonding, in which first is this and second is this. Okay. Third is ionic bond. Fourth is hydrogen bond. Fifth is the van der Waals forces, or we can say here fifth is the hydrophobic bonds. Understood? Majority of proteins and enzymes present in the protoplasm exhibit tertiary structure. You have to remember that majority of proteins and enzymes for their functioning, okay, they are having protoplasmic or in the present in the protoplasm have tertiary structure. Okay. So tertiary structure, it is a structure in which the amino acids, the proteins, they get folded very much to form a globular appearance. They are water soluble, okay, and they form colloidal solution. This structure has following bonds: the peptide bond, which is the strongest bond, then the disulfide linkage, then the ionic bonds, then the hydrogen bonds, then the hydrophobic bonds. Disulfide linkage is present in between sulfur and hydrogen of amino acids, especially the cysteine, mean the sulfur containing amino acids. Okay. Ionic bond. The formation of ionic bond between the two opposite ends of a protein molecule is due to the dispersal of charges or electrostatic attraction between them. Majority of proteins and enzymes present in protoplasm, they exhibit tertiary structure or they have tertiary structure, right? This is the tertiary structure. The hydrogen bond you can see between hydrogen and oxygen. Okay. This is the ionic bond between COO minus and NH3 plus. This is the ionic linkage, ionic bond. Okay. These are the hydrophobic bond of a hydrophobic chain alkyl group. This is disulfide linkage. Disulfide linkage. These are van der Waals forces of interaction. Okay, van der Waals forces of interaction. Okay. This is the third I was telling about, the collagen helix. The collagen helix. Okay. So when we talk about the tertiary structure, so it becomes very important to know about a very important term, denaturation and renaturation. What exactly denaturation is? It is the process of degrading the tertiary structure. Process of degrading the tertiary structure. It is a process of degrading the tertiary structure when pH, optimum temperature, okay, any heavy metal intoxication, heavy metal intoxication. So when pH is abruptly changed, optimum pH is changed, optimum temperature is changed, okay, heavy metal intoxication, okay. So these changes, they lead to degradation of the tertiary structure, and this process known as what? Denaturation. So because of the process of denaturation, the protein, it is it loses the biological activity, loss of biological activity. It can coagulate or precipitate, or precipitation happens. Understood? And if the denaturating agent is removed, then it re-establishes the bonds, okay, to maintain the tertiary structure. That is known as what? Renaturation. Okay. So this is the tertiary structure, and the process of degrading of tertiary structure is known as what? Denaturation. pH, temperature, heavy metal intoxication, okay, then radiations. There are many factors which helps to control the, or which are responsible for maintaining the structure of a protein. And if it is lost or if it is degraded anyhow because of these reasons, then it is the part of denaturation. It is known as what? Denaturation. During denaturation, the protein loses its biological activity. Okay, it no longer remain functional for performing that particular work. Quaternary structure. The polypeptide chains that have tertiary structure are linked by different bonds to form a quaternary structure of a protein. There are different polypeptide chains with similar, like lactic dehydrogenase, or dissimilar types, hemoglobin and insulin. So polypeptide chains having tertiary structure, they come up with more, formation of some more bonds, formation of domains. Okay, domains is like a one micro unit having multiple polypeptide chains having primary, secondary, and tertiary structure. So these are different polypeptide chains having similar or dissimilar types of structures like lactic dehydrogenase or hemoglobin or insulin. Quaternary structure is the most stable structure. It is the most stable structure of a protein. Okay. So quaternary structure, it is found in multimeric proteins only. Multimeric proteins found in multimeric proteins. Okay. Understood? Like hemoglobin is having four polypeptides, 2 alpha and 2 beta. Okay. Myoglobin, hemoglobin, insulin, lactic dehydrogenase, like that. Okay. Now, what are the different properties? These are the levels of protein structure: primary structure, secondary structure, already explained, tertiary. This is the quaternary structure. You can see the quaternary structure. Okay. Properties. The very first property is specificity. Specificity means each spe species has certain specific proteins not found in the others. Okay, mean every species having some specific proteins belonging to their origin. Okay, so there are some specific proteins which are belonging to particular origin of particular species. They are not present in the others. Okay, so closely related species, they have some common proteins, some common proteins. That is why they share the characteristics, similar characteristics. Okay, so number of common proteins decrease with an increase in dissimilarity between species. Okay, so as we change the species type, the number of common proteins decreases. The principle is used to bringing out evolution or evolutionary relationship. So this is known as what? Setting a evolutionary relationship with the biochemical evaluation, that is the enzymes and all. Then they are colloids. Okay, they are not completely soluble in water. They are colloids. Okay. Then reactivity, as they have intramolecular and intermolecular hydrogen bonding and they are folded, protein comes to have a particular surface configuration with specific reactive groups. They have specific reactive groups on their surface. Okay, because of their specific reactive groups on the surface, their reactivity changes. The specificity provides functional diversity to proteins for performing different cellular activities. Like enzymes are specific due to their proteinaceous nature. Antibodies are complex glycoproteins which are especially very specific to particular antigen because of their proteinaceous nature. Okay. So reactivity, they have reactive groups on their surface, and these groups are very specific for binding, like in case of antibodies, they bind to a specific toxin or antigen. Permeation, as they are macromolecular in size, they are not permeated through cell membrane. As they are macromolecular in size, they are not permeated through cell membrane. Okay, that is why they move through exocytosis and endocytosis. Okay, they are amphoteric. They are also amphoteric. Denaturation already explained. They are macromolecules when they have larger size. Okay, their size or their mass, their molecular weight ranges from 4,500 to 4.6 million. 4,600 in case of adrenocorticotrophins to 4.6 million. Pyruvate dehydrogenase. Pyruvate dehydrogenase. Their molecular weight can vary up to a large extent. They are very big in size. Okay, they are macromolecules. So, my dear students, this is all about the properties and all. Okay, now we are going to discuss about the types of proteins. The types of proteins are categorized as simple, compound, and derived. Simple proteins, they are only made up of amino acids, then compound, and then derived. Simple, made up of only amino acid like fibrous proteins. They are long, coiled, and thread-like collagen, like elastin. Collagen, it maximum levels are present in the animal body. One third part of the total proteins is collagen present in the connective tissue. It helps to make thread of collagen and known as tendons. Elastin, it is also connective tissue, thread of elastin known as ligament. Okay, and keratin. So collagen, elastin, and keratin. Collagen, it makes tendons. Elastin, it makes ligaments, and keratin. Okay, so they all are fibrous proteins. Globular proteins, which help in transportation, like rubisco, present in the chloroplast in plant cells, most abundant protein present on the earth. Albumin, it maintains the blood colloidal osmotic pressure in milk as lactoalbumin, in egg as ovoalbumin, in blood as serum albumin. Okay. Globin, which is present in hemoglobin, helps in the transportation. Histone proteins present with eukaryotic DNA. In it, lysine and arginine amino acids are present in more amount in histone proteins. Protamine, which is present in nucleus of sperm. Okay. Prolamines present as stored protein in cereal grains, in barley as hordein, in maize as zein, in wheat as gliadin or gluten or gluteline. So the globular proteins and the fibrous proteins, they are the simple proteins which are made up of only amino acid. There is no other group which comes in the conjugation with this, like fibrous and globular. Fibrous, they are long, coiled and thread-like. Then globular, fibrous, it includes collagen, elastin and keratin. Collagen, this is the maximum protein which is available, okay, in the body. then elastin, and then keratin. Okay. Globular, it includes rubisco, albumin, globulin, histone proteins, protamines, prolamines. Okay. Prolamines, they are stored proteins which are present in different grains like maize as zein, barley as hoden, wheat as glutenin. Protamine, they are present in nucleus of sperm. Histone proteins, they are present in the eukaryotic DNA. Lysine and arginine, they are majorly found in the histone proteins. Globin, it is present as a hemoglobin, okay, to transport the oxygen and nutrients. Albumin, it maintains the blood fluid osmotic pressure. In milk as lactoalbumin, in egg as ova albumin, and in blood as serum. So compound and derived proteins we will be discussing in the next lecture. Till then, bye bye, take care, stay safe, stay